This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Thank you, Aaron. So it's my pleasure today to introduce Walter Tsang. Walter is a Bachelor of Veterinary Science Honours graduate from 2015 from the University of Queensland. And he's worked as a veterinarian in private practice for some years and has also done a bit of other types of work as well, including in the Department of Agriculture and Fisheries in the Queensland Government and a bit of work in a diagnostic lab at UQ as well. So Walter joined us as a PhD candidate in 2021, and he started remotely from Brisbane because Brisbane was not in lockdown and Melbourne was very much in lockdown. So Walter started remotely, but then he was able to join us here in Melbourne at the start of this year. And since then, he's been very busy in the lab working on his project. So I'll turn it over to Walter to tell us all about his project. Thank you.
Thanks, Jo. Welcome everyone to my PhD confirmation seminar. I want to start off by acknowledging the traditional owners of the land on which we're meeting today, and I pay my respects to their elders, past, present, and emerging. So my topic is on infectious laryngotracheitis virus, or ILTV in short. My supervisors are Jo, Carol, Turgot, and Amir. So I want to start off with a bit of introduction on ILTV. So it's a species of alphaherpesvirus, which is a subfamily under the family of herpesvirus. It causes respiratory clinical signs characterized by nasal discharges, sometimes hemorrhagic nasal discharges, dyspnea, which is difficulty breathing, coughing, and mortalities. The virus is transmitted by aerosol or expectorant, and it causes significant production and therefore economic loss in the poultry industry. The control of ILTV is mainly by vaccinations and biosecurity measures, which we'll get into. So a bit on the pathogenesis of ILTV. The main routes of entry for ILTV in birds are upper respiratory tract and conjunctiva of birds. And it replicates in the epithelium of nasal cavities, trachea, larynx, and conjunctiva, and it causes pathology in the trachea. The virus can disseminate to different organs in infected birds, and it undergoes latency in trigeminal ganglion and trachea. By undergoing latency, it avoids recognition by immune system, and the latent infection can then be reactivated by stresses. A little bit on the history of ILTV. So the virus itself is first discovered in 1925 in the US. And since then, different classes of ILTV can be identified using different genetic materials from different genes. So class 1 to 5 have previously been identified prior to 2007 in Australia. Since then, class 2 and from there class 9 and 10 have been reported, and class 2 was the previously most prevalent classes, and now that has been replaced by class 9 and 10. There's a linkage between class 9 and 10 and concurrent use of Australian and European ILTV vaccine strains, which I'll get into in a second. But the key message from that is that ILTV evolves over time, and that changes with the disease that it can cause in the industry. So vaccination for ILTV. Most of the vaccines used in Australia are live attenuated vaccines. So Zoetis is the manufacturer for two of them. One of them is the strain A20, the other one is the strain SA2. Both of these can be given via eye drop or drinking water methods. And the third one is made by MSD, and it's the service strain of the vaccine, and that can be given in an eye drop method. These vaccines are effective in controlling clinical cases, but not viral replications in infected birds. And this has an implication on vaccine safety because, number one, there's a high frequencies of recombination occurring between and in the virus strains. And secondly, although these vaccines are attenuated, they're not attenuated in a traditional sense. So in an incorrect or uncontrolled dosage of vaccines that are given to the birds, that can cause the birds to get actually sick from the virus, from the vaccine itself, and it can cause adverse effects, and therefore vaccine safety can be affected by that. A little bit on recombination. So recombination is the main evolution pathway for ILTV. Like I said before, ILTV as a species changes constantly. Recombination occurs between two different strains, and that was first reported within APCA in 2012. And since then, frequent recombination in both field and laboratory settings have been described and reported. So recombination basically is when there's co-infection of two different strains of ILTV in one host cells and causing mixing and exchanging of virus genetic material or recombination of the genome sequences, causing a hybrid new strain. And there's a potential for new recombinant virus strains to have a higher level of virulence and transmissibility. And again, because of inappropriate vaccine usage, that can cause recombination in this scenario as well. So to summarize all that, my project focus is to examine the emergence, distribution, persistence, and transmission of ILTV recombinant viruses in the natural host species, which is chicken, and determine how vaccination practices influence these processes. There are three main aims of my project. The first one being to describe viral and epidemiological characteristics of an outbreak of infectious laryngotracheitis. The second one is to characterize ILTV recombination and the effect of drinking water vaccination in specific pathogen-free chickens, or SPF chickens, in an experimentally setting where they're co-infected with two ILTV field strains. And aim number three, in vivo characterization of key recombinant viruses in SPF chickens. So for aim one, to describe an outbreak. So this particular outbreak is from a broiler chicken farm in Victoria. It's a commercial broiler chicken farm. It has around 43 to 44,000 birds per shed, and it has 7 sheds in total within the same property. And the information provided by this property is in the form of mortality data in an Excel sheet, and different samples that were submitted. So there were 3 tracheal samples submitted in formalin and 4 tracheal samples submitted in viral transport media. So starting with the data set. So again, the outbreak was from a broiler chicken farm in Victoria. The outbreak occurred in September 2021. And this is an example of the Excel sheet that I got from this property. So we can see here the age is the number of days that the birds have been in this particular sheds. Mortality and culls, so natural mortality and culls, the birds that have been culled for the reasons that are listed here. And this is the total daily mortality in this column. And each of the 7 sheds basically have their own table. And the vaccines that were used on this property is the A20 vaccine in drinking water form, given on day 13 and day 21 here. So from there, using this data, the graph that was plotted, first off, I want to just put it in a graphical sense or content to see how it looks like in a chart. So this is a daily mortality rate of chickens per shed. Each of the lines in different color represents the daily mortality rate of each shed, as I've written here. This axis here is the number of days of these birds in this property. And then this here is the daily mortality rate in percentage. So we can see there's an initial spike in daily mortality rate, and that is somewhat expected due to the age of the hatchlings being moved into a large group and the stress of being relocated itself. And so that is somewhat expected. And then after that, it calms down a little bit. Like I said, vaccines given on day 13 and day 21. Beyond that, given the clinical signs that the producer was noticing at the time here, as well as the spike in mortality, that would be the actual onset of the outbreak of ILTV. So the producer was noticing dyspnea, coughing, and sort of the hemorrhagic nasal discharges we've mentioned before. So starting at this point, we're starting to see a spike in the daily mortality rate. And from there, a cumulative mortality rate graph was made. And so again, this is in each of the sheds in different colors. This is still the number of days of the birds in the sheds. And this, again, is the percentage cumulative mortality rate we can see here. So from the initial data set, the data that was used to get to this point was the number of days in production, starting population, and then the total mortality in the form of daily culls plus the mortalities. And the reason why these numbers were made is to estimate the R0 value. So the R0 value is the basic reproduction number. That's the average number of individual and initial case can infect in a population with no immunity. So we can see here there's a little steeper part in the cumulative mortality rate, and that part would be the actual outbreak of ILTV within this property. It starts off at different rate, and we can see here the earliest shed seems to be starting here, and then that kind of spread across the entire property. And this property already uses all the recommended biosecurity measures. So we can see even with this biosecurity measures in place, the virus can still effectively cause an outbreak within the entire property. From there, to estimate the R0 value, linear regression of the natural log of each of the cumulative curve was made only at the part of the exponential growth phase, so the steeper part of the curve we saw on the slide earlier. And then the slope of each line then represents the epidemic growth rate. So we can see here, this is the actual days in the period of exponential growth, so the steeper part of the cumulative curve, and this is the natural log of the with linear regression in place. And so in this equation here, y equal to 0.05x, the number before the x is the slope or the growth rate of the epidemic, and each of the shed has their own slope value as well. So each of them has their own growth rate. And then the R0 value of each shed was then calculated using this expression here. I'll explain the expression a little bit because there's a lot of symbols there. So L and D represents the average duration of the latent and infectious periods, respectively. Latent period in this case is not the latency I was talking about before in regards to herpes virus or ILTV. Latent period in this scenario means the incubation period for the outbreak. So from the time point of the virus infecting the birds to when they actually starts to be infectious and showing symptoms. Lambda is the growth rate or the slope at which exponential growth occurred. M and N are gamma distribution parameters, and therefore allowing assumptions for latent and infectious period to be varied, meaning that within this infected class, there can be subdivisions. M and N were assumed to be 100 to allow for a tight distribution around the mean in this estimation. So like I said before, the epidemic growth rate or the slope of those linear regressions of each shed here are shown here, and the mean of them is around 0.076. And from there, it was estimated with the latent period to be assumed at 2 days, and infectious period to be assumed between 2, 4, and 6 days. The reason why these assumptions were made for the infectious period is because looking at birds that are infected experimentally, peak infection is usually on day 4. And by day 6, all of the birds that were tested positive would have been tested positive on day 4 as well. So beyond that, peak virus level would be falling down already. So infectious period was assumed to be 2, 4, and 6 days in my estimation. And we can see here, these are the R0 value in each of the sheds. Shed 1 to shed 4, fairly similar. So 1.18 to 1.35 in that range. Shed5 and Shed7 seems to have a higher level, so 1.28 to 1.73 in the case of Shed7. Overall, the mean are not value depending on the assumption on the infectious period duration, ranges from 1.25 to 1.46. Comparing that to a study that was done also in Victoria in 2011, the R0 value was 1.98 to 2.93. And those data in this particular study was from 2008 to 2010. Again, from the outbreak that I've looked at, the R0 value was from 1.25 to 1.46. The difference between these two studies, keeping in mind, is the one in 2011, the birds were unvaccinated, whereas the birds in 2021, they're vaccinated, although both of these two groups of birds are from a field setting. Keeping that in mind, moving on to the other samples that were submitted within this outbreak in 2021. So there were samples that were submitted in formalin. These samples were sectioned and then H&E stained to assess them in a histopath setting. So histopathological scoring system of ILTV was established in 1990 in this study, and it ranges from grade 0 to 5. Grade 0 being normal and grade 5 being severely affected. So this is an example of how a normal trachea would look like in a cross-section under histology. So this is the wall of the trachea, and this here would be the lumen of the trachea itself. Here we can see a nicely arranged layer of epithelial cells. So they are tightly packed with glandular tissues or glandular cells in between, and they each have little cilia on top of them to help move mucus within the trachea lumen. So the grading system here that was described, it's based on the loss of epithelial structure, infiltration of Repler cells within the epithelial layer, leukocytes or inflammatory cells infiltration, and the sloughing of the epithelium away from the lamina propria layer and the cartilage layer here. So with the sample that have been stained from this particular outbreak in 2021, we can see that there's massive infiltration of leukocyte or inflammatory cells, so the darker cells here, and mucosal hemorrhage, so infiltration of Repler cells within the mucosal layer. It's not showing sloughing of the mucosa yet, but we're seeing the loss of the glandular tissue before. And zooming in as well, we can also see some syncytia, so cellular damage and cellular death within the mucosal layer as well. And so from there, the histopath changes were graded to be a grade 3 using the scoring system. Moving on to other samples, they were submitted in the viral transport media. So these trachea sample were from 4 birds. They were submitted again in VTMs. Unfortunately, the samples weren't identified to belong to different sheds in itself. They just come as samples from 4 different birds. These samples were homogenized and inoculated in choroallantoic membrane or CAMs of embryonated SBFX. So just to explain a little bit with that, this is an egg with an embryo inside. So CAM would be a membrane that's within the eggshell itself. And the goal of the CAM inoculation and the process is to separate the layer away from the eggshell using negative pressure. And then from there, the sample were then inoculated onto the CAM themselves for a period of around 5 days or so. After that, after 5 days, so come time of harvesting, we can see that there are little pocks formed on the CAM themselves. So this little white circle we can see on the CAMs are little pox formation indicating cellular infection, likely by the virus in this case. And we can see that on another CAM as well, again, on these little white circles on there. the goal of which is to isolate the virus within these tracheal samples. Each of these CAMs were then homogenized, and that had gone through a viral quantitation step. So first, I wanted to confirm that there's presence of ILTV within these samples. So the viral load were measured using a UL15 primer qPCR just to confirm that there's a positive results from these samples, and there were. So these samples were then re-inoculated into a new set of CAMs as a step of viral passage and amplification. And a total of 16 CAM samples were then made, and they were again aliquoted as well. These 16 samples were then quantified with the UL15 primer qPCR again to assess the viral load within these samples. And I was able to achieve viral load more than 10 to the power of 7 genome copy numbers per mil from these samples. And 4 of these samples from the original 4 tracheal samples were then sent for whole genome sequencing. So these samples were sent to Charles River for whole genome sequencing. The data was analyzed using Genius Prime. Processes that have been completed so far were trimming, assembling with reference to class 9 genome of ILTV, and they've been aligned against other Australian ILTV strains as well. The analysis of the sequence is currently still under progress, but what I'm trying to aim for is to see how it correlates with the other strains. So both previously different classes of ILTV as well as the vaccine strains that have been used in Australia. So to summarize the part 1 or aim 1 of my project, so to describe an outbreak of ILTV the outbreak was in a vaccinated commercial broiler chicken farm in Victoria, and the outbreak was in September 2021. The R0 value from this outbreak of ILTV was 1.25 to 1.46. There were histopathological changes consistent with ILTV infection noted in this outbreak. Virus, also the ILTV virus, have been isolated, and currently sequence analysis is in progress. Anecdotally, and also with data provided from this producer, they're using a newer vaccine protocol. So they're using a vaccine called VacSafe in drinking water form. It is showing a much lower growth rate of the virus. So I was unable to estimate the R0 value from this newer data set because there was not even a distinguishable exponential phase. So I couldn't actually calculate the actual slope, or I can't separate the time frame. So that concludes kind of the aim 1 of my projects. Moving on to aim 2. So just a reminder, the second aim is to characterize ILTV recombination and the effect of drinking water vaccine in SPF chickens that are experimentally co-infected with 2 ILTV field strains. So this is an in vivo experiment conducted by Turgid in 2021. And looking at recombination between 2 field strains of ILTV. So the 2 field strains that were used were CSW1 and V199. And this is the timeline of the experiment. So we're looking at co-infection, well, first vaccinating these birds in different vaccine strategies, and then co-infecting them with the 2 strains I've mentioned before. The aim or the goal of the experiment is to look at how different vaccine strategies might affect or change the evolution of ILTV in the form of recombination. These birds were vaccinated in a drinking water method as well. So these data somewhat can be comparable to the field outbreak as previously described. And like I said, the goal is to compare viruses that are replicating in vaccinated birds versus birds that are not vaccinated altogether, or birds that are vaccinated in a suboptimal kind of setting. And these samples were then used to look at recombination in the form of using a TACMAN recombination genotyping assay to look for specific genetic variations of each strains, and from there to detect recombination in the progeny of viruses after mixed infection. So far, the samples from this in vivo experiment, parts that are completed are the DNA extractions from these tracheal swabs, qPCR, again, just to confirm and quantify the viral levels in these samples, and plaque purification and picking to isolate these viruses. The next step would be to complete the Taqman genotyping process to actually see what recombination we are looking at. And from there, there are going to be a few selected recombinant viruses that will undergo full genome sequencing as well. So for what's next in my project for aim number 1, so for the outbreak description, would be to complete sequence analysis of the viruses that were isolated, and then examining these viruses in vitro, looking at the growth kinetics of them. Looking also at the recombination patterns of these viruses, and from there, it will be utilized in aim 3, which is to in vivo characterize these viruses, both looking at effects in virulence, transmissibility, pathogenicity, and just to see how they do behave in vivo. For aim 2, we're trying to compare the difference in recombinants detected in vaccinated versus unvaccinated birds. And again, for aim 3, in vivo and in vitro assessment of tissue tropism, virulence level, transmissibility, and pathogenicity by using the dominant recombinants from aim 2 and looking at how they do behave. So to conclude, that didn't work. ILTV is ever evolving in a way of recombination, investigation in how transmissibility and virulence can be affected by vaccination strategies is very important in controlling the disease, and that has a significant social economic impact. That concludes my seminar. I want to say thank you to my supervisors, Joe, Carol, Turgut, and Amir, my chair, Andrew, and special thanks to Matthew, Denise, Mauricio, Andreas, and Gaia for the help along the way as well. Thanks, everyone.
Thank you, Walter. That was a really interesting talk. Very happy to turn over to the audience for any questions or comments or thoughts. I'm happy for people just to unmute themselves and ask questions, or alternatively, people might like to put something in the chat, perhaps. While people are thinking of questions, I can think of a couple of questions. So, Walter, you've got sort of 2 streams, I think, in your PhD project. You're looking at field outbreaks, or at least one field outbreak of ILTV compared with your experimental sort of controlled conditions in experimental birds. How well do you think that those 2 different streams can be compared? Do we think the experimental work is a close mirror of what goes on in the field, or do you expect that there's a few differences between experimental infection and field infection?
There's going to be quite a bit of difference, unfortunately, between field and experimental setting. Obviously, in field, there's not a lot of things you can control. And so with introduction of different strains of ILTV in a field setting, I think, yeah, you can't really control exactly what comes into play or what strains is available for the mixing in that sense. But I think with experimental setting, it gives us an insight into how different strains can be affected by vaccine strategies. And then that is also a knowledge we can then apply to the field setting. Even though it's not exactly identical, the principle should stay the same.
Great, thank you. No, that's a good answer. Any other additional questions from audience?
We have some well wishes in the comment section saying a great presentation and fantastic talk, Walter. Great talk. Best wishes for your PhD work.
Thanks, Corinne. Thanks, Abdul. Thanks, Matthew.
Great. Well, thank you, everyone. So I think we'd just like to thank everyone for coming along today. And obviously Walter's talk was very clear and not too many questions required. So thank you for coming. Thank you, Aaron, for organising everything. And of course, thanks and congratulations to Walter for his talk. Thanks, everybody.
Thanks, everyone.